Alright, back into Chapter 6. This is the second part. We're going to be looking at the organelles of the cells. To start with, the nucleus. The nucleus works in conjunction with the ribosomes, as the nucleus contains the DNA. Recall DNA is used to make RNA in the nucleolus, which we see right here. The RNA can leave the nucleus, going through some of these nuclear pores here, where the RNA will now be in the cytoplasm. The RNA will then go to the ribosomes, and then the ribosomes take the RNA and convert it into a protein. The nucleus contains most of the cell's genes, and is typically the easiest one to point out, the easiest organelle to point out underneath the microscope. We saw that especially true with those staining techniques previously. The nuclear envelope encloses the nucleus, which separates it from the cytoplasm. The nuclear envelope is a double membrane, and we can see this in the image above. You can see the outer layer, actually it's right there, outer membrane, and then the inner membrane. This double membrane is a lipid bilayer, just like the cell membrane we spoke of once again a little earlier. Once again, there are pores. Those are on the outside of the nuclear membrane, which allow for things to either get into or out of the nucleus. We spoke of the RNA being able to sneak through those. Next we have ribosomes, which we had already spoken a little bit about. They're made up of rRNA, or ribosomal RNA, and protein. I guess it should be mentioned that our DNA in the nucleus can make 3 different forms of RNA. The first, ribosomal RNA, or rRNA, messenger RNA, also called mRNA, and transfer RNA, or tRNA. We'll get into the nitty-gritty of these later. Back to ribosomes. They can be attached to the endoplasmic reticulum or free-floating in the cytosol. Regardless of location, cytosol or on the endoplasmic reticulum, both are used to make proteins or protein synthesis. Going back here, we can see some of these ribosomes studded to the exterior of the endoplasmic reticulum. Now, some of the organelles in the cell are considered to be part of the endomembrane system, and I'm sure you're going to recognize a lot of the names on this list here. These organelles work together, hence they're a system. They communicate or send things back and forth, either directly as they can be attached to one another, or through the use of vesicles. You could think of vesicles as tiny enclosed bottles that carry messages around the cell. The first on our list is the nuclear envelope, which we spoke of as it surrounds the nucleus. It has a lipid bilayer, and it has pores for things to get into or out of the cell. So we're going to move into the endoplasmic reticulum, which we can see is continuous with the nuclear envelope right here. The endoplasmic reticulum has 2 distinct regions, the rough ER and the smooth ER, rough endoplasmic reticulum, smooth endoplasmic reticulum. Recall that ribosomes are attached to the rough ER, hence the name rough ER. It has a roughened appearance, and we can see that going on over here, a little bit of rough, and then over here where there's nothing, it's smooth. Now, the presence or absence of these ribosomes give the endoplasmic reticulum itself different functions. For example, the smooth ER synthesizes lipids, metabolizes carbohydrates, detoxifies poisons, and stores calcium. The rough ER, on the other hand, makes glycoproteins. Recall glycogen is a carbohydrate, so glycoproteins are proteins with carbohydrates bound to them. The rough ER also distributes transport vesicles. Once again, recall vesicles are used to transmit messages, send things around the cell. These vesicles are proteins themselves with a membrane. And speaking of membranes, the rough ER also produces the items necessary for constructing cellular membranes, whether it's the cell membrane or the membranes for the membrane-bound organelles themselves, such as the nucleus. The third component on the list of the endomembrane system was the Golgi apparatus. You may have heard of the Golgi as being the cell's post office. This is because it modifies products of the ER, packages and sorts materials into the transport vesicles we just spoke of. And there are two phases to the Golgi, the cis and trans phases. We have the cis phase face back here, the trans phase up in the front part. The cis side, or cis face, will be the receiving end, and this is where vesicles come to. And you can see it's actually carrying something. It's binding to the cis face of the Golgi apparatus, and it looks like these two little purple dots are going to get into the internal spaces of the Golgi apparatus, which we can see is kind of folded out like this and flattened out, forming a shipping vesicle on this side that will be sent elsewhere. So it is like a post office. It takes things in, modifies them, sorts them, stores them sometimes, and then ships them out to other destinations. Something you should maybe start thinking about is how this shape of this Golgi apparatus fits its function. You're going to need to know this with most of the organelles in the cell here. So let's look at it. We can see that it goes back and forth in these tiny little passages, and then all of a sudden we have something being shipped out. Start thinking about, and I'm not going to tell you the answer to this, how does the shape of this fit the function of vesicles dumping off products and then being shipped out to other organelles? So start thinking about those types of things. The next component are the lysosomes. Lysosomes are quite small but are very powerful. Lysosomes are membranes of enzymes that can digest macromolecules, or nutrients, the proteins, the fats, the polysaccharides, even nucleic acids, DNA and RNA. Lysosomes play a role in the intake of food into the cell, and we can see this going on in the left-hand image above. Phagocytosis or phagocytosis is where food will approach the membrane of the cell. The cell will form a little vesicle by pinching off around the food particle, and then the food's now in the cell, the protein, the carbohydrate, whatever you happen to have. And then lysosomes will attach themselves to that food vacuole, and then these digestive enzymes will start kind of attacking sort of the food and break it up into smaller substances. Much easier for the cell to make use of the smaller particles than the larger ones. These enzymes within the lysosome are also used for a process called autophagy. This is essentially the removal of organelles from the cell. Sometimes organelles get kind of worn out and they need to be replaced before they start producing waste products for the cell and cause an increase in toxins. And when these organelles do, they, once again, just like the food over here, they are enclosed in the vacuole for safekeeping. Lysosomes come along, they have this double membrane. It attaches to the vacuole with the non-functional organelles at this point, and it breaks them up, and then these products are able to get out of the cell before they become a larger problem for the cell. Once again, form meeting function. Start thinking about how these tiny lysosomes, double membranous capsules, and the enzymes inside, how does the form and shape of the lysosome fit the function of what it does? Once again, start thinking about those things. Vacuoles. Vacuoles can have several different functions, as there are several different types of vacuoles. Food vacuoles are formed during phagocytosis. We saw those on the previous slide, and how the cell membrane pinches off a vacuole that surrounds the food. There are also contractile vacuoles. Contractile pump. So contractile vacuoles will pump, specifically water out of the cells. Then there are the central vacuoles. We see these in the plant right here, a little bubble. And there's it in the real cell, kind of large. These central vacuoles hold organic or carbon-based compounds. They also hold water, and they work sort of as a storage system, kind of like a water tank in a town, if you will, or maybe here in Arizona, the canals. And again, form meeting function. Start looking and thinking about the shapes of vacuoles and how they're shaped and structure and how they're put together. How does that fit the function of what it is they do? Alright, so recapping on the endomembrane system, we have the nuclear envelope, once again surrounding the nucleus. Certain things will leave the nucleus through these little pores in the nuclear envelope and go into the ER, which, as you can see, surrounds the nucleus mostly. The ER has several different functions, produces these little vacuoles. The vacuoles then go to the Golgi, specifically the cis face of the Golgi, where the contents are emptied. They go through a series of channels and then pop off at the end in a vesicle, which can fuse to one of these lysosomes and then be removed from the cell. So, putting it all together. So this concludes part 2 of the tour of the cell. Part 3 we'll start looking at other organelles that we have yet to talk about, mitochondria, chloroplasts, energy, so on and so forth. So I will see you in part 3.